Good morning. It's February 12th, and this is your daily brief in science. Here's everything you need to know. A multinational study led by Flinders University has identified a molecular trigger for VITT, showing that an adenovirus PVII protein can induce autoimmune antibodies against PF4, which can lead to rare clotting events. Using mass spectrometry sequencing, researchers demonstrated molecular mimicry and pinpointed the adenovirus vector itself as the trigger, not a specific vaccine component. Experts say robust vaccine safety surveillance remains essential to maintain public confidence and guide safer vaccine design and risk mitigation. The research acknowledges international collaboration and diverse funding, including the Deutsche Forschungsgemeinschaft, the Gates Foundation, and the European Medicines Agency, supporting sequencing resources and work. The findings extend prior work from Australia, Germany, and Canada, noting uniform antibody characteristics across patients and structural analyses of VITT antibodies. While the risk of VITT is extremely rare, the study offers a blueprint for designing safer vaccines and broader protection against disease. Previous genetic associations, notably IGLV 3.21*02, are integrated with proteomic data to support a unified model of susceptibility and mechanism. The research opens pathways for improved diagnostics and preventive strategies, enabling earlier detection and a precision vaccinology approach that monitors autoimmune risks in vaccine design. Government compensation programs related to VITT payouts have drawn scrutiny over eligibility and adequacy, though claims processing continues. The study is the culmination of NEJM published work tracing VITT from early recognition to identifying the molecular trigger, with potential to translate into safer, widely accessible vaccines. Case context emphasizes human impact, recalling 8 Australian deaths from VITT during the rollout, underscoring the debate over messaging versus safety. Analyses suggest that modifying or removing the adenovirus protein could prevent VITT while preserving vaccine effectiveness, offering a roadmap for safer future adenovirus-based vaccines. A study from Silent Spring Institute tested 43 hair extension products, both human hair and synthetic, and found hazardous substances in many samples. About 1 in 10 samples had organotin compounds above European safety limits, and several chemicals detected are linked to cancer, early puberty, and metabolic issues, with Black women and girls disproportionately affected due to higher product use. The findings point to a need for stronger regulatory oversight and more transparency in the hair extension industry to protect consumer health. Industry voices call for better guidance for consumers and professionals, noting extended scalp contact and gaps in information about chemical treatments. Stakeholders emphasize labeling gaps and a lack of ingredient transparency that hinders informed choices and professional guidance on scalp safety. The study notes a lack of federal safety standards, testing requirements, or recalls for beauty products. With only limited state bills in California and Washington and no comprehensive United States regulation yet, regulatory developments show a patchwork. The Food and Drug Administration has faced delays in banning certain formaldehyde-related hair treatments, while the Environmental Protection Agency has proposed loosening restrictions on carcinogens, signaling uneven protections. Some mitigation ideas emerged, such as pre-washing synthetic hair with apple cider vinegar to reduce tin-based chemicals, though this does not fully eliminate exposure. The broader implication is a push for mandatory ingredient disclosures, testing for sensitizers, and wider regulatory reform to ensure safer, affordable options for consumers. Advocates urge stronger government regulation and clearer ingredient disclosures rather than relying on consumer vigilance to ensure safety. Legislators in New York and New Jersey are considering bills to require ingredient disclosure and to restrict harmful chemicals, while a federal Safer Beauty Bill package seeks to boost Food and Drug Administration oversight of synthetic hair products. The study's authors advocate for stronger regulation and disclosure. Several states are moving toward bills, and the Safer Beauty Bill package at the federal level would expand oversight of synthetic extensions. A new setback for Moderna as the Food and Drug Administration issues a refusal to file letter saying the new mRNA flu vaccine application isn't ready in its current form because the trial comparison did not use the best available standard of care. Chief Executive Officer Stefan Bancel says the company will push for a comprehensive review with an FDA-approved comparator and stresses transparency to clarify the path forward. The move comes amid a broader shift in United States health leadership that could influence vaccine policy and federal research priorities under the current administration's circle. Analysts are watching whether the administration's stance on vaccines and compounding will raise regulatory pressure on companies and telehealth platforms, potentially impacting patient access and industry practices. Experts warn that a restrictive regulatory direction could have wider implications for the mRNA platform and future vaccine development. Mid-sized biotech firms are signaling concern and solidarity in response to proposed drug pricing policy, highlighting industry-wide anxiety about policy shifts. Regulatory actions around compounding include FDA warnings, a Justice Department investigation request, and a Novo Nordisk patent dispute against Hims and Hers, pointing to a tightening environment for compounded products. Some note the letter's unusual signing by the center's director and question alignment between staff recommendations and the final decision, with inputs from agencies. Other items in the issue touch on broader healthcare policy moves, AI and healthcare discussions, and industry moves like Lilly's acquisition of Orna Therapeutics. The full article is behind a Stat Plus subscription and requires access for complete analysis. The Stat Plus paywall limits broader readership from seeing the full context and quotes. A team of scientists warns humanity may be closer to a point of no return than previously thought, with runaway global heating risking a hothouse Earth and severe climate impacts. Oregon State University researchers say tipping points could destabilize the climate system, triggering rapid warming and sea level rise if thresholds are crossed. A new paper argues the risk of a hothouse Earth is growing as warming accelerates and could set off a cascade of climate tipping points. Expanding renewable energy, protecting carbon-rich ecosystems, and adopting climate-resilient, socially just policies to phase out fossil fuels quickly are among recommended responses. Forests are shifting from carbon sinks to sources due to fire and drought, while oceans are losing part of their capacity to absorb CO2. Key signs include ice sheet instability, faster warming rates, and changes in carbon cycle dynamics across forests and oceans. Permafrost thaw releasing methane and carbon dioxide could reinforce warming through feedback loops. Experts cited include Dr. Christopher Wolff, Prof. Johann Rockström, and Prof. Hans Joachim Schellenhuber. Prof. Tim Lenton and Prof. William Ripple led the study in One Earth, noting AMOC weakening and Amazon dieback could amplify warming and interact with other feedbacks, underscoring insufficient current climate action. Delaying action narrows opportunities to avert severe and unmanageable climate outcomes. Current warming likely exceeds the Paris Agreement 1.5-degree target based on recent temperatures, though formal determinations use a 20-year average. A new gene editing approach aimed at dramatically lowering LDL cholesterol is showing early promise, but experts caution that long-term safety and durability remain unknown. The strategy targets liver edits to turn off ANGPTL3 or PCSK9, with early trials reporting about a 50% drop in LDL and triglycerides at higher doses within roughly 2 weeks. Researchers emphasize that these edits are permanent, so rigorous large-scale testing is essential before any clinical use. Initial studies are conducted by Verve Therapeutics and CRISPR Therapeutics, with trials in Australia and the UK and US sites opening, as next steps are planned later this year. The trials use a single infusion to deliver the gene editing tool, seeking to identify optimal targets, dosing, and safety while enrolling individuals at very high cardiovascular risk. While the approach aims to reduce risk, guidelines for immediate cholesterol control continue to target LDL levels around 70 mg per deciliter for high-risk patients and 100 mg per deciliter for healthy individuals, using statins and approved therapies. Observations draw on natural mutations that inactivate ANGPTL3 or PCSK9 as a blueprint for potential outcomes, but permanent edits demand careful long-term safety evaluation. Even if gene editing proves viable, established heart health practices remain critical, including lifestyle changes, blood pressure and diabetes management, and traditional cholesterol-lowering therapies. Statins and other standard treatments remain the backbone of cholesterol management, though adherence issues and residual risk fuel interest in new approaches. Trials prioritize enrolling those at very high cardiovascular risk to better understand unknowns and long-term effects of the edits. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.